Molecular biology is a growing field in science, and the ability to manipulate macromolecules can greatly enhance our lives. A concept scientists are experimenting with is biomolecular chirality. What is biomolecular chirality, also known as handedness? To put it simply, biomolecular chirality deals with molecules that are mirror images of themselves but aren't superimposable. These mirror image molecules are called enantiomers. Let's try to visualize this. A good example of something chiral is your own hands. For example, if you try to stack your own hands on top of one another, they are not superimposable. The same is with many 3D molecules. Take a simple amino acid, for example. We have an amine group, a carboxyl group, and a side chain labeled R. Now let's create a mirror image of this amino acid. As you can see, they are not superimposable, although they have the same bonds and arrangement of atoms. To separate these molecules, one is called left-handed, or L for short, and one is called right-handed, or D for short. But what makes this molecular property biologically significant? They are basically the same, right? Although they may be very similar in structure, they interact with other molecules and organisms very differently. One example is a drug called thalidomide. This drug was marketed and sold to pregnant women as a cure for morning sickness. While one hand of the drug did cure morning sickness, the other hand caused severe limb disformations in a developing fetus. Since this was not known, and the drug was made in relatively equal amounts of each hand, which resulted in what BBC called as one of the biggest medical tragedies of modern times. So now we know that these chiral molecules have different properties when compared to their other hands. But what other biological significance is there? Another observation that has been made is that most amino acids in nature are left-handed, while most sugars are right-handed. This suggests that nature prefers one of two hands instead of an equal distribution of both. So now that we know that most of life is one-handed, the question of creating life on the other hand emerges. Would it be possible to create a mirror image of an organism, or even a human, all the way down to the molecular level? If we could, this opens up a plethora of possibilities. For example, we know that pathogens detect a potential host or target by recognizing proteins or other molecules that are synonymous with living organisms. These pathogens are able to invade the host using specific interactions with things like the cell membrane, which it's able to do because of specific proteins it has evolved to interact with. An organism that has been mirrored all the way down to the molecular level will have the opposite hand for all molecules within it. This would enable a mirrored organism to be immune to almost all pathogens, as they wouldn't even be able to recognize it as a potential host. This pathogenic resistance could be very useful to humanity, although bioterrorists could potentially synthesize mirrored pathogens. However, there is a downside to all of this. Most food products would not provide any nutritional value to these mirrored organisms, as the mirrored enzymes wouldn't be able to properly interact with molecules such as glucose, which would then require the production of a bunch of mirrored foods. Another useful application could be in bacterial engineering. Scientists can manipulate mirrored bacteria and reverse engineer them into non-mirrored bacteria once a desirable trait has been created. This would allow scientists to carefully control growth and production of these mirrored bacteria, as well as eliminating the possibility of a biohazard if a bacteria were somehow able to escape the lab. This is due to the fact that a mirrored bacteria wouldn't be able to get any nutrition from its environment and would die quickly. Hopefully I was able to teach you something about biomolecular chirality and maybe even inspire you to become a molecular biologist. Thank you for watching.